Hello viewers, today let us learn something about ultrastructure of bacterial cell. Myself Dr. Madhuri from Government Degree College, Hussainiali. This topic is having different contents such as definition, shapes and different components of cell and bacterial growth, nutrition, reproduction as well as economic importance. Now let us see the definition of bacteria. Bacteria are prokaryotic microorganisms which are single celled and has circular DNA in it. Prokaryotic means, pro means primitive and karyon means nucleus. In these cells, the circular DNA is not bound by nuclear membrane. In such condition, the chromosome, bacterial chromosome is called as nucleoid. Bacteria also lacks membrane bound organelles such as endoplasmic reticulum, mitochondria, etc. They are showing different types of plasmids which are rendering different functions for the bacterial cell, and the size of bacteria is often measured in micrometers. Now the interesting question is, where are these bacteria found? Bacteria are actually omnipresent. It means they can be seen everywhere wherever you want to see them. They are present in the air, soil, water, on your body, inside your body, everywhere. And these are the earliest inhabitants of the earth, around 3 × 10⁹ years old. Bacteria are showing symbiotic and parasitic relationships with plants as well as animals. It is interesting to know that if you take 1 gram of soil, there can be 40 million bacteria in it. And if you take human body, whatever be the number of cells in human body, 10 times more number of bacteria are present in your body. Now, what is the importance of bacterial studies? Why we have to study about bacteria? Bacteria, due to their smaller size and short reproduction time, they are used as models in different fields of research. They are used in fermentation industries. They are helping us in nutrient recycling and also in nitrogen fixation. Nitrogen fixation is the conversion of atmospheric nitrogen into ammonia. About 90% of nitrogen fixation is done by bacteria. Not only this, they are also used as food for animals and humans. They are used for cleaning environments, recycling of waste, minerals, and even they are responsible for soil fertility. This slide, you can see the size of bacteria, where it stands. This is the eukaryotic cell, the size of which is varying from 100 to 100 micrometers to 1 mm, and lesser to that is organelles which are present in the eukaryotic cells, and bacteria is even smaller than these organelles. The size of bacteria can be between 1 micrometer to 10 micrometers. As we have discussed, they are very smaller in size. The spherical bacteria is showing size of 0.5 to 2 microns, and rod-shaped bacteria is showing 1 to 10 microns in length and diameter of 1.25 to 1 microns. Spirochetes are spiral cells which are measuring about 500 microns in length. The bacteria which you can see through naked eye is Epilopicium fischeloni. It is measuring about 600 microns in length and 80 microns in diameter. Without microscope, you can see this bacteria. The world's smallest bacteria is Mycoplasma gallisepticum. It is measuring around 200 to 300 nanometers. And the world's largest bacteria recently discovered is Thiomargarita namibiensis. This bacteria is measuring around 0.1 to 0.3 millimeter, that is 100 microns to 300 microns. Sometimes the size of this bacteria can go up to 0.75 mm, that is 750 micrometers. Coming to the shapes of bacteria, bacteria exhibits different types of shapes. First one is cocci, that is spherical bacteria, bacillus, rod-shaped bacteria, vibrio, comma-shaped bacteria, and spirillum is spiral-shaped bacteria. Now we'll study each shape in detail. The spherical bacteria, they are showing different types of arrangements, that is, they can be present in a single cell form, that is micrococci. It is seen in Micrococcus roseus, and diplococci, where two cells are together, and example is Diplococcus pneumoniae. The next arrangement is streptococci. Here the cells are arranged in a chain form. This kind of arrangement is due to the division of cells in a single plane. After division, the cells are remaining together, giving a chain shape. This kind of cells are seen in Streptococcus lactis. Next is Staphylococcus. Here the cells are arranged in a grape bunch form. This kind of arrangement is due to the division of cells in three planes but in irregular fashion. Then comes tetrad, where four cells are arranged together in a packet form. Example is Aerococcus and Pediococcus. And the last is Sarcina, where 8 cocci are staying together in a form of cuboid manner. This kind of arrangement is seen in Sarcina lutea. Sarcina kind of arrangement is due to division of cells in three planes. Next shape is Bacillus. The Bacillus also show different types of arrangements. The word Bacillus has come from Latin word bacillum, which means stick or rod shaped. In this, there are also cells called Cocobacillus. Cocobacillus are actually, they are intermediate between cocci and bacillus. Many times these kind of cells are mistaken to be cocci. Such kind of cells are seen in Haemophilus influenzae. The bacilli can be single cells, example Lactobacillus, or diplobacilli, where two cells are together, example Corynebacterium diphtheriae, and Streptobacilli, where chain of cells are together, which is seen in Streptococci moniliformis, and palisade kind of arrangement, where the cells are arranged parallel to each other like matchsticks in a matchbox. Such kind of arrangement is seen in diphtheria bacilli. Next is Vibrio. Vibrio is nothing but a comma-shaped bacteria. It is seen in Vibrio color. Then comes our Spirillum. These are spiral-shaped bacteria. They appear like corkscrew. They are found in Treponema pallidum. These were, whatever we have discussed till now, are the basic shapes. Now, apart from these shapes, there are also other shapes such as filamentous bacteria, example Candidatus Savagella, rectangular bacteria, example Halorcula vallis mortis, star-shaped bacteria which are found in Stella, and the last is pleomorphic bacteria which can be seen in Mycoplasma pneumoniae. Pleomorphism is a characteristic where the cells can change their shape for a short time or temporarily. Now we will see the components present in the bacterial cell. Here in this slide we can see the structure of a bacterial cell, where from the outside there is a capsule. Beneath this there is a cell wall, cell membrane, cytoplasm, and on the outer side there are flagella, pili. Inside the cytoplasm there is a nucleoid and plasmids and ribosomes. All these structures we have to study one by one. Coming to the first structure, that is flagella, which are present on the outer side of the cell. Flagella is a plural form and the singular form is flagellum. This flagellum is made of proteins called as flagellins. They are often used as taxonomical tool to classify the bacteria, and if you see the structure of the flagellum, it consists of three parts, that is basal body, hook and filament. The main function given by flagella is motility. It can be of different types. In this slide we can see the structure of flagellum. As we know, the cell wall in gram-positive cells and gram-negative cells is different in their construction. Here in this slide, the right side is the gram-positive cell and the flagella is having only two rings, that is M ring and S ring. M ring stands for membrane ring and S ring stands for supermembrane ring. Whereas in gram-positive, gram-negative cells there are four rings, that is M ring, S ring, P ring. P ring is peptidoglycan ring and L ring is lipopolysaccharide ring. In gram-negative bacteria there are four rings in the basal body. Now let us see the flagellar arrangement. Based on the flagellar arrangement, there are different types of cells. If a cell is having a single flagella, it is called as monotrichous. If it is having a tuft of flagella on one side of the cell, then it should be called as lophotrichous. If there are two tufts of flagella on either side of the cell, then it is called as amphitrichous. And if many flagella are present on the entire surface of the cell, it is called as peritrichous, and in the cells where flagella is absent, it should be called as atrichous. These are the examples where different types of flagella are seen. Monotrichous flagella is seen in Pseudomonas aeruginosa. Lophotrichous is seen in Pseudomonas fluorescens. Amphitrichous is seen in alkaligens and peritrichous is seen in Salmonella typhi. Coming to the next appendage is pili or fimbriae. These pili or fimbriae are usually seen in gram-negative bacteria. They are made of protein called pilin. They are smaller, shorter and more in number than flagella. Pili are longer than fimbriae. The production of pili is controlled by genes present on plasmids, and the pilin protein consists of 163 amino acids. Pili do not help in motility of the cell, but if we remove these pili from the cell, it can form them rapidly again. There are 5 to 6 types of pili which are giving different functions to the cell. Coming to the functions of pili, it possesses antigenic property, like whenever the bacteria enters the host system, the host immune system can raise antibodies against the pili. Fimbriae agglutinates with blood cells. They are also affecting metabolic activity of the cell. Sex pili are used for the transfer of genome by the process called conjugation, and some pili are also used for attachment of the bacteria to surfaces. Our next component is capsule or slime layer. Bacteria usually synthesize a slimy or gummy material outer to the cell wall. This is generally called as glycocalyx. If this glycocalyx is tightly bound to the cell, it is called as capsule, and if it is loosely bound, it is called as slime layer. The main function rendered by capsule is pathogenicity. Pathogenicity is the ability of the bacteria to cause the disease. Capsule can be made of polysaccharides or polypeptides. Polysaccharide capsules are seen in Streptococcus xanthomonas, and polypeptide capsules are seen in Bacillus subtilis and Bacillus anthracis. These are the images where we can see the capsule. It is seen as a halo around the bacterial cell. It usually repels the stain, and it is stained by gram-negative staining method. Coming to the functions of cell capsule, first and the foremost function is pathogenicity or virulence, and it protects the cell from desiccation as it contains high amount of water. It helps the cell in excluding bacteriophages and other toxic chemicals. It helps the cell to attach to the streams, plant roots, teeth, tissues and other bacteria. These are species specific. And capsule is also acting as buffer between cell and external environment. Our next component is cell wall. It is rigid layer covering the cell, and it gives protection to the cell from osmotic pressure, and the main function is giving shape to the cell. The thickness of cell wall ranges from 100 to 250 angstroms. Based on the reaction or response of a bacteria to gram stain, the bacteria are recognized as two types. They are gram-positive cells and gram-negative cells. In the cell wall, the main component is peptidoglycan. This peptidoglycan consists of three parts. One is backbone, second one is tetrapeptide side chain, and the third one is peptide interbridge or cross-link. Now coming to the first part, that is backbone. The backbone of peptidoglycan consists of repeating and alternating units of amino sugars called as NAG and NAM. NAG is N-acetyl glucosamine and NAM is N-acetyl muramic acid. Both these amino sugars are linked by beta-1,4 glycosidic linkage. Coming to the next component, that is tetrapeptide side chain. This tetrapeptide side chain consists of a small peptide of 4 amino acids: L-alanine, D-glutamic acid, diaminopimilic acid and D-alanine. The composition of this tetrapeptide is different in gram-positive cells and gram-negative cells. The sequence of amino acids or the composition of tetrapeptide which is shown here is of gram-negative cells, and most of the gram-positive cells will have L-lysine in the third position, that is in place of diaminopimilic acid, L-lysine is present in gram-positive cells. Coming to the next component, that is peptide interbridge or cross-link. The adjacent layers of peptidoglycan should be linked to each other. This linkage or the cross-link is done between the peptides of adjacent peptidoglycan layers. This linkage or the cross-link is different in gram-positive and gram-negative cells. In gram-positive cells, for example, in Staphylococcus aureus, the cross-link is by pentaglycine bridge. It means 5 glycine amino acids are joining 2 strands of tetrapeptides. Whereas in gram-negative cells, the linkage between 2 tetrapeptides is between the amino group of diaminopimelic acid and carboxyl group of alanine. Here we can see the difference between gram-positive cell wall and gram-negative cell wall. Till now we have discussed about peptidoglycan layers. As we can see in the picture, the peptidoglycan is more in gram-positive cells and it is very less in gram-negative cells. In gram-positive cells, apart from peptidoglycan, there are teichoic acid molecules. Teichoic acid is nothing but they are the polymers of glycerol or ribitol which are joined by phosphate groups. The teichoic acid, it can be bound to lipids of plasma membrane or it can be bound to peptidoglycan layers. If the teichoic acid molecules are bound to lipids, then it is called as lipoteichoic acids, and if it is bound to peptidoglycan, then they are called as cell wall teichoic acids. Apart from teichoic acids, there are other polysaccharide molecules. In gram-negative cells, the construction of the cell wall is very complex when compared to the gram-positive cells. Here in gram-negative cells, there is a periplasmic space. Periplasmic space is the space between plasma membrane and the outer membrane. Here there is a very less content of peptidoglycan, and outer side of the peptidoglycan there is a outer membrane followed by lipopolysaccharide membrane. The permeability of outer membrane is due to presence of porins. Here in this slide we can see the differences between gram-positive and gram-negative cells. In gram-positive cell, there is more amount of peptidoglycan, whereas in gram-negative cells there is less amount, and cell wall has relatively less amino acids in gram-positive, whereas in gram-negative they are having more amino acids. And teichoic acids are present in gram-positive cells and are absent in gram-negative cells. Lipopolysaccharides are absent in gram-positive and are present in gram-negative cells. Gram-positive cells are sensitive to penicillin and gram-negative cells are sensitive to streptomycin. Coming to the functions of cell wall, the main function is to maintain the shape of the cell. It is acting as a protective barrier which is allowing molecules into the cell up to a particular size. O antigen, which is present in the lipopolysaccharides of gram-negative cells, is showing antigen specificity. Matrix proteins are acting as receptors for nutrients, colicins and bacteriophages. It protects the protoplasm from mechanical damage and from osmotic rupture or lysis. Cell wall is the site of enzymes for respiration, photosynthesis, lipid synthesis, etc. Our next component is plasma membrane. The plasma membrane is present beneath the cell wall. It comprises of about 8 to 15% of cell's dry weight. The thickness of plasma membrane is 5 to 10 nanometers. It is actually a lipoproteinaceous membrane consisting of 70% proteins and 30% lipids. In this figure, we can see the plasma membrane. Here, there is a lipid bilayer. The lipid consists of hydrophilic head and hydrophobic tails. So, these lipids are arranged in such a fashion that hydrophilic heads are on either side, covering the hydrophobic tails inner to the membrane. In this lipid bilayer, there are proteins embedded. Proteins are of two types, peripheral proteins and integral proteins. Peripheral proteins are soluble proteins and they can be dissociated from the membrane, whereas integral proteins are non-soluble and they can't be dissociated from the membrane. There are also some oligosaccharide molecules which are present in the membrane, and apart from this there are hopenoids. Hopenoids are pentacyclic sterol-like molecules. They are synthesized from the same precursor as steroids and they provide stability to the plasma membrane. Coming to the functions of plasma membrane, they help in transportation of nutrients. They contain enzyme systems for different mechanisms. It is a site of electron transport and oxidative phosphorylation, and it is also acting as specific attachment sites for chromosome and plasmid replication. Coming to the next structure, that is mesosomes. Mesosomes are nothing but the invaginations of plasma membrane, usually found in gram-positive bacteria. These mesosomes are thought to play a role in cell wall formation during cell division, chromosome replication, or as a site of oxidative phosphorylation. But recently, scientists have found similar folds in bacteria which are subjected to particular class of antibiotics and antibacterial peptides. So, they have concluded that these mesosomes, they may not be the part of bacterial cell and they may be formed during some special conditions for protecting the cell. Next components are ribosomes. As we know, bacteria consists of 70s ribosomes. Ribosome comprises of rRNA and proteins. rRNA is around 60% and proteins are around 40% in the ribosome. 70s type of ribosomes in prokaryotes consists of two subunits, that is 50s and 30s subunits, whereas in eukaryotes there are 80s type of ribosomes consisting of 60s and 40s subunits. S stands for Swedberg units. The diameter of ribosome is around 200 angstroms. And in bacterial cells we can see polyribosomes. Polyribosomes are nothing but aggregates of ribosomes seen in the cytoplasm. These ribosomes aggregate around the mRNA for translation or protein synthesis. In this picture, we can see the electron micrograph of ribosome. The blue area is 50s subunit and the pink is 30s subunit along with RNA molecules. Here also there is 30s subunit, 50s subunit, and they together consist of 70s ribosome. Next important component which is present inside the cytoplasm is nucleoid. As we have discussed, in prokaryotes there is a single circular double-stranded DNA. It is distributed in an irregular pattern in the cytoplasm. This chromosome is not covered by any nuclear membrane. That is why it is called as nucleoid. Nucleoid can be stained by Flagella stain and can be observed under compound microscope. The DNA is found to be attached to the cell membrane, which may help in replication of cells. Next is proteins. Nucleoid consists of 60% DNA, 30% RNA and 10% proteins. The proteins present in nucleoid, they are resembling the histones, but they are named as HU, NS and DNA binding protein 2. The important characteristic of nucleoid is they lack introns as well as histones. Introns. Usually eukaryotic DNA consists of coding sequences and non-coding sequences. Coding sequences are called as exons and non-coding sequences are called as introns. In bacteria, that non-coding sequences are absent. E. coli size is 2 to 6 micrometers. This small cell is keeping the DNA of 1400 micrometers length in it. How it is possible? Such a long DNA, it undergoes extensive supercoiling to get fit into that smaller cell. The supercoiling is by turns. The one more important genetic material or the DNA molecules inside the cytoplasm is plasmids. These are extrachromosomal DNA molecules. The size and number of plasmids varies from cell to cell. They help the cell in getting protection from antibiotics or stress conditions. Plasmids can be transferred from one cell to another by mechanism called conjugation, and this process is utilized in rDNA technology. The number of plasmids vary from 1 to 100. If the number of plasmids is less, they are called stringent plasmids, and if it is more, they are called as relaxed plasmids. Sometimes plasmids integrate into the main chromosome, then it is called as episome. Plasmids are of different types: F plasmid, R plasmid and Col plasmids. In this picture we can see the plasmids. Coming to the functions of plasmids, they are used for making toxins, they resist environmental factors, they are also used for resisting antibiotics, and they can degrade unusual chemicals. Now coming to the specialized cell structures. In specialized cell structures, first one is storage granules. As we store food in our home, bacteria also storing some kind of food materials in its cell. First one is here granules. These granules are composed of specific substances such as glycogen, polyphosphate or sulfur granules or PHB. In this case, the granules are not covered by any membrane. Next is gas vacuoles. These gas vacuoles are aggregates of gas vesicles and they are permeable to atmospheric gas. These are membrane bound structures which are present in aquatic photosynthetic bacteria and cyanobacteria. They help the cell to regulate the depth at which they float, which in turn helps in photosynthesis. Next is carboxysomes. Carboxysomes are the organelles which are helping the cells in carbon dioxide fixation. These are seen in cyanobacteria, nitrobacter and nitrosomonas, and they contain enzyme RUBP carboxylase. They can be of 2 types, alpha carboxysomes and beta carboxysomes. Next is magnetosomes. In 1975, Blackmore has for the first time shown that bacteria are having magnetosomes. These magnetosomes consist of magnetite. They are seen in Aquaspirillum magnetotacticum. They show magnetotaxis. Interesting feature here is this kind of magnets are seen in heads of birds, dolphins, tuna, turtles, etc., which are aiding them in navigation. Here in this picture we can see a series of magnets arranged in a linear form and showing North Pole and South Pole. Next important structure is endospores. As we know, these are formed during unfavorable conditions or whenever the supply of carbon, nitrogen, phosphorus is limited, then gram-positive cells will go for sporulation. Spores can be seen in the genus of Bacillus, Clostridium, Sporolactobacillus, Sporosarcina, etc. These are used for resisting heat, radiations, chemical disinfectants, desiccation, etc. Endospores can survive in boiling water for several hours, whereas vegetative cells can be killed at 80 degrees for 30 minutes. Coming to the shape and position of spore. Spores show different shapes such as oval, ellipsoidal and spherical. And they can also show different positions such as terminal, subterminal or central position. Spores are having DNA, RNA, enzymes and calcium dipicolinate. Apart from this, there is 10 to 30% of water and there are some proteins called as small acid soluble spore proteins, SASPs. The main function of SASPs is to bind to DNA and protect it from UV, desiccation, dry heat. They also serve as carbon and energy sources during the time of regeneration. In this picture we can see the different spore positions and here the construction of the spore. Outer layer is exosporium, below that is cortex, below that is spore coat and then cell wall is present and core or cytoplasm is there. Whenever the conditions are favorable, the spore will germinate and vegetative cell is released. Due to presence of tough membrane, tough spore coat, the spores will not allow any stain to enter into it. So there is a special mechanism to stain the spores. It is done by using malachite green. In this staining method, the counter stain is safranin. At the end of the staining, you can see pink colored vegetative cells having green colored spore in it. Coming to growth, bacteria usually divides by binary fission where the cell number doubles for every division. Generation time ranges from 15 minutes to few hours and it may vary for a same bacteria with nutritional and environmental conditions. This is the growth curve showing different phases. Coming to the reproduction. Reproduction of bacteria can be of asexual or sexual types. Asexual reproduction is by binary fission or endospore formation and sexual reproduction is by conjugation, transduction or transformation. Transformation is the uptake of nascent DNA from the external environment and conjugation is the transfer of genetic material from one cell to the another by forming conjugation tube. Transduction is the transfer of genetic material by bacteriophages. And coming to the nutrition, aspect. Depending on the light energy and the chemical energy, there are different types, phototrophs and heterotrophs. Coming to the economic importance, bacteria are showing different beneficial activities, such as they are helping us in biogeochemical cycles and in agriculture by nitrogen fixation, and in various industries they are used like food industry and silk industry or leather industry. Many functions are being done in these industries, and medicine for antibiotic production as well as in biotechnology, they are used for production of insulin using rDNA technology. Not only beneficial activities, these bacteria also harm us by spoiling our food and by denitrification, which leads to loss of fertility, and they are also causing diseases like in plants. We can see different diseases such as blight of rice, citrus canker, and in humans cholera, typhoid, tuberculosis, etc. And in animals anthrax and TB can be seen. We have learned different concepts such as size and shape of the cells, flagella and pili, which are present exterior to the cell wall, and capsule, the structure of cell wall and plasma membrane, and what is nucleoid, and the plasmids and ribosomes we have seen, and the inclusion bodies such as carboxysomes, storage granules, vacuoles, magnetosomes, and endospore we have studied, and we have learned about growth, nutrition, as well as economic importance of bacteria. With this, I take leave. Thank you for patiently listening.